Hey there, welcome back to the channel. In today's video, we'll be breaking down some important GCSE biology topics: type of cells, subcellular structures, and microscopy. I'll walk you through everything you need to know in a way that's easy to understand. So let's dive in. First, let's start with the two main types of cells: eukaryotic and prokaryotic cells. These cells are the foundation of all life, but they're different in some important ways. Eukaryotic cells include both animal and plant cells. They are bigger and more complex and have a nucleus to store DNA. The nucleus is like the control center of the cell, holding all the important information it needs to function. On the other hand, prokaryotic cells are smaller, simpler, and are found in bacteria. They don't have a nucleus, instead, the DNA is in a single loop, and they often have little rings of DNA called plasmids. These plasmids are like small bonus pieces of genetic info that can help bacteria survive in certain environments. Now, let's take a closer look at the subcellular structures that make up these cells. These are the parts inside cells that help them function. In eukaryotic cells, both animal and plant cells share some important structures, but plant cells have a few extra. Let's go through them. Nucleus. This is found in both plant and animal cells. It contains the DNA and controls the cell activities. Cytoplasm. Found in all cells. It's a gel-like substance where most of the cell's chemical reaction takes place. Cell membrane. This surrounds all cells, controlling what enters and leaves the cells. Mitochondria. These are the powerhouses of the cell where energy is produced. Ribosomes. These are the protein factories where proteins are made. In addition to these, plant cells have a few unique features, like cell wall and chloroplast. Cell wall. These provide support and structure. It's made of cellulose and is found in plant cells only. Chloroplast, found only in plant cells. These contain chlorophyll, which helps plants make food through photosynthesis. Finally, prokaryotic cells, like bacteria, also have some important structures. They have a DNA loop, which is a single strand of DNA, and may also have plasmids. Next, let's talk about microscopy. To study cells and their structures, we use different types of microscopes. There are two main types of microscopes: light microscopes and electron microscopes. Light microscopes are the ones most commonly used in school. They use light and lenses to magnify samples. They're great for looking at larger structures like the nucleus of a cell. However, they have lower magnification and can't show really tiny details like organelles. On the other hand, electron microscopes uses beams of electrons, which have a much higher magnification and resolution. This allows scientists to see much smaller details like ribosomes and mitochondria, but the downside is that these microscopes are more expensive, requiring more preparation, and the samples need to be dead. Now let's tackle something a little bit more mathematical: the magnification formula. This formula is super useful when you're using a microscope to look at cells and need to know how much the image has been magnified. The formula is simple: magnification equal image size divided by the actual size. Here's an example. A student observes a plant cell using a light microscope. The image of the cell measured 25 millimeters across on the microscope slide, but the actual size of the cell is 0.025 millimeters. So we just substitute the values into the formula. Magnification equal 25 divided by 0.025, which is equal to 1,000. So the magnification used in this case is 1,000 times. Next, we have a required practical activity: observing cells under a light microscope. Let's walk through the steps of preparing the slide to look at plant and animal cells. Step 1. You prepare the slide. First, you need to place a small drop of water on a clean microscope slide. Step 2. You add a specimen. Place the specimen on the slide. This could be a piece of onion skin or cheek cells. Step 3. Stain the specimen. To make the cells easier to see, add a drop of iodine solution. Iodine stains the cells, helping you see the structure clearly. Step 4. Cover the specimen. Gently place a cover slip over the specimen to avoid air bubbles. Step 5. Microscope observation. Start with the lowest magnification on your microscope to locate the cells, and then switch to a higher magnification for a much more detailed observation. Step 6. Drawing. Finally, use the pencil to draw the cells you see, adding labels and the scale bar to show the size. That's it. You're now ready to observe and draw cells for your practical. Before we finish, let's go some review questions. These are great practice for your exam, so pause the video if you want to. Now, let's go over the answers to the review questions. Make sure you've written down your responses before checking these. That's a wrap for today's video. If you found this video helpful, don't forget to like, subscribe, and hit the notification bell so you never miss a video. If you have any questions, leave them in the comments below. Thanks for watching, and I'll see you in the next video.